Our next experiment is estimation of transpiration using an apparatus called Porometer. Let's see transpiration. Transpiration is a biological process by which water is lost in the form of water vapor from the aerial part of the plants. Why transpiration is important for plants? Because it helps in the absorption and upward movement of water and minerals from the root to the leaves and also prevent the plants from heating up. That is, it is a primary determinant of the leaf energy balance and plant water status. Water is passively transported to the root and then to the xylem. The forces of cohesion and adhesion causes the water molecules to form a column in the xylem. The water moves from the xylem into the mesophyll cells of the leaves. Then it evaporates from the surface and the leaves and leaves the plants by diffusion. This apparatus is called porometer. Porometer measures the amount of water being evaporated from a leaf through small pores in the leaf known as stomata. This is a null balance porometer. That is, when we keep a transparent leaf inside a chamber, it loses water. That changes the relative humidity inside chamber. In order to maintain a constant relative humidity inside chamber, we passes a dry airflow to the chamber. The null balance porometer based on the measurement of dry airflow rate necessary to maintain a constant relative humidity inside a chamber when a transparent leaf is placed. That is, the water lost by the leaf can be measured by means of null balance porometer. The null balance porometer that we are using for measuring the transpiration is a Lycor model 1600 is a steady state porometer. It is the porometer that operating in null balance principle. According to this principle, the apparatus gives direct values of stomatal resistance calculated from the relative humidity, leaf and air temperature, and flow rate measured values. The equation used by Li 1600 to obtain leaf transpiration rate is E. The leaf transpiration rate is equal to Pc minus Pa into F by A. E. It is the leaf transpiration rate which is expressed in milligram per centimeter square per second. And Pc is the water vapor density in the chamber which is expressed in milligram per centimeter cube. And Pa is the water vapor density in the dry air flow entering in the chamber. It is assumed as 2% of the relative humidity. And F is the volumetric flow rate in the chamber. And A is the leaf area. It is expressed in centimeter square. As shown in this equation, the calculated leaf transpiration rate is directly depends on the volumetric flow rate into the chamber. And this flow rate depends on the chamber temperature and barometric environmental pressure. The Lycor 1600 porometer has two main parts, that is console and also a leaf chamber. And this is the schematic representation of Lycor 1600 porometer showing the different parts of porometer. First one is the console. It has some control elements that make possible the transpiration measurement. The pump that draws air from inside the console and pumps it to the air tank where the air is depressed in two directions. One part escapes via the internal flow control. The internal flow control that controls and adjusts the relative humidity inside the chamber to a set point. And the other part goes to null adjustment valve that limits the null adjustment valve limits the maximum flow rate entering in the chamber and therefore forces a major airflow to escape from the system. This depression is essential in the system that leaves with different transpiration. The leaves with different transpiration rate must be measured and controls on the flow rate entering in the chamber will be necessary to maintain the relative humidity in the set point. For example, a leaf is placed in a chamber has a very low transpiration rate. Only a small dry air flow rate will be necessary into the chamber to balance the leaf transpiration rate. If air entering into the system is too high, the relative humidity can drop below the set point and the measurement will not be real. Therefore, the system has the portion of venting of excess flow. Once the excess flow passes through the null adjustment valve, it will pass through two desiccants. One in the console, that is this is one desiccant, and the other on the sensor head. This is the sensor head and this is one sensor and the other one is on the console. We have one desiccant here and also on the sensor head. So we ensure that the airflow entering in the chamber is dry. And this is the chamber. The airflow entering into the chamber is dry by means of these two desiccants. One on the console and on the other on the leaf sensor head. The second part, leaf chamber or sensor head. Transpiration depends on many factors. Therefore, the leaf which is measured must be normally enclosed in a chamber where major number of possible external factors can be controlled. In the same way, the size of the selected chamber must permit the measurement of different leaves with very different sizes, but also with different stomatal resistance. Therefore, a high variety of chambers are available for measuring transpiration. Cylindrical chamber of Li 1600, for example, is made of nickel plated aluminium to minimize the water absorption. The sensor inside the chamber check for several environmental parameters. For example, leaf and air temperatures, relative air humidity, and also PAR, that is photosynthetically active radiation. Once the dry air flow entering the chamber, it mixes with the water vapor lost by the transpiring leaf, and the dry air flow rate will be necessary to maintain the balance the relative humidity inside the chamber. This value is the key for the later stomatal resistance and transpirational calculations. This is regarding the measurements. The first step to use Li 1600 is to check that the desiccants from the console is completely dry. Every week the desiccant from the sensor head must be changed. Once that the sensor head is cleaned without the presence of pollen or dust, the apparatus is ready to work. Because the dust of pollen which can increase the water vapor absorption with unusually increased values. For starting the work with Lycor 1600, a previous habituation to the environment parameters is necessary, which is about 30 minutes. Therefore, we must be careful to install the instrument in the definitive measuring place. Direct sun must be avoided to protect the chamber against the abrupt temperature changes. At this temperature, checking checking of chamber and the leaf temperature, flow, relative humidity, PAR, etc. can be done. Once finished the acclimatization and after calibrating relative humidity sensor and setting the null point humidity to ambient relative humidity and the dry airflow rate entering into the chamber, data recording can start. Along this time, we can display the parameter values of our interest. Li 1600 needs generally a mean time of 10 to 20 seconds to reach a steady state situation on a leaf with diffusive resistance of 5 seconds per centimeter. With higher values, the instrument needs a longer time. Once the instrument is at or near the null point relative humidity and the diffusive resistance value is stable, data can be stored in a data memory and transferred to a PC with a data transfer program for later analysis.